Color vision is the ability of an organism or machine to distinguish objects based on the wavelengths, or frequencies, of the light they reflect, emit, or transmit. Colors can be measured and quantified in various ways. Indeed, a person's perception of colors is a subjective process whereby the brain responds to the stimuli that are produced when incoming light reacts with the several types of cone cells in the eye. In essence, different people see the same illuminated object or light source in different ways. Wavelength and hue detection. Isaac Newton discovered that white light, after being split into its component colors when passed through a dispersive prism, could, by passing them through a different prism, be recombined to make white light. The characteristic colors are, from long to short wavelengths, and, correspondingly, from low to high frequency, red, orange, yellow, green, blue, and violet. Sufficient differences in wavelength cause a difference in the perceived hue. The just noticeable difference in wavelength varies from about 1 nm in the blue-green and yellow wavelengths, to 10 nm and more in the longer red and shorter blue wavelengths. Although the human eye can distinguish up to a few hundred hues, when those pure spectral colors are mixed together or diluted with white light, the number of distinguishable chromaticities can be quite high. In very low light levels, vision is scotopic. Light is detected by rod cells of the retina. Rods are maximally sensitive to wavelengths near 500 nm, and play little, if any, role in color vision. In brighter light, such as daylight, vision is photopic. Light is detected by cone cells which are responsible for color vision. Cones are sensitive to a range of wavelengths, but are most sensitive to wavelengths near 555 nm. Between these regions, mesopic vision comes into play and both rods and cones provide signals to the retinal ganglion cells. The shift in color perception from dim light to daylight gives rise to differences known as the Poirconier effect. The perception of white is formed by the entire spectrum of visible light, or by mixing colors of just a few wavelengths in animals with few types of color receptors. In humans, white light can be perceived by combining wavelengths such as red, green, and blue, or just a pair of complementary colors such as blue and yellow. Physiology of color perception. Perception of color begins with specialized retinal cells containing pigments with different spectral sensitivities, known as cone cells. In humans, there are 3 types of cones sensitive to 3 different spectra, resulting in trichromatic color vision. Each individual cone contains pigments composed of opsin apoprotein, which is covalently linked to either 11-cis-hydroretinal or more rarely 11-cis-dehydroretinal. The cones are conventionally labeled according to the ordering of the wavelengths of the peaks of their spectral sensitivities, short, S, medium, M, and long, L, cone types. These 3 types do not correspond well to particular colors as we know them. Rather, the perception of color is achieved by a complex process that starts with the differential output of these cells in the retina and it will be finalized in the visual cortex and associative areas of the brain. For example, while the L cones have been referred to simply as red receptors, microspectrophotometry has shown that their peak sensitivity is in the greenish-yellow region of the spectrum. Similarly, the S and M cones do not directly correspond to blue and green, although they are often described as such. The RGB color model, therefore, is a convenient means for representing color, but is not directly based on the types of cones in the human eye. The peak response of human cone cells varies, even among individuals with so-called normal color vision. In some non-human species this polymorphic variation is even greater, and it may well be adaptive. Theories. 2 complementary theories of color vision are the trichromatic theory and the opponent process theory. The trichromatic theory, or Young-Helmholtz theory, proposed in the 19th century by Thomas Young and Hermann von Helmholtz, as mentioned above, states that the retina's 3 types of cones are preferentially sensitive to blue, green, and red. Ewald Hering proposed the opponent process theory in 1872. It states that the visual system interprets color in an antagonistic way, red versus green, blue versus yellow, black versus white. Both theories are now accepted as valid, describing different stages in visual physiology, visualized in the diagram on the right. Green, magenta and blue-yellow are scales with mutually exclusive boundaries. In the same way that there cannot exist a slightly negative positive number, a single eye cannot perceive a bluish-yellow or a reddish-green. But such impossible colors can be perceived due to binocular rivalry. A range of wavelengths of light stimulates each of these receptor types to varying degrees. Yellowish-green light, for example, stimulates both L and M cones equally strongly, but only stimulates S cones weakly. Red light, on the other hand, stimulates L cones much more than M cones, and S cones hardly at all. Blue-green light stimulates M cones more than L cones, and S cones a bit more strongly, and is also the peak stimulant for rod cells, and blue light stimulates S cones more strongly than red or green light, but L and M cones more weakly. The brain combines the information from each type of receptor to give rise to different perceptions of different wavelengths of light. The opsins, photopigments, present in the L and M cones are encoded on the X chromosome. Defective encoding of these leads to the two most common forms of color blindness. The OPN1LW gene, which codes for the opsin present in the L cones, is highly polymorphic. A recent study by Varelli and Tishkoff found 85 variants in a sample of 236 men. A very small percentage of women may have an extra type of color receptor because they have different alleles for the gene for the L opsin on each X chromosome. X chromosome inactivation means that while only one opsin is expressed in each cone cell, both types occur overall, and some women may therefore show a degree of tetrachromatic color vision. Variations in OPN1MW, which codes the opsin expressed in M cones, appear to be rare, and the observed variants have no effect on spectral sensitivity. Color in the human brain. Color processing begins at a very early level in the visual system, even within the retina, through initial color opponent mechanisms. Both Helmholtz's trichromatic theory, and Herring's opponent process theory are therefore correct, but trichromacy arises at the level of the receptors, and opponent processes arise at the level of retinal ganglion cells and beyond. In Herring's theory, opponent mechanisms refer to the opposing color effect of red-green, blue-yellow, and light-dark. However, in the visual system, it is the activity of the different receptor types that are opposed. Some midget retinal ganglion cells oppose L and M cone activity, which corresponds loosely to red-green opponency, but actually runs along an axis from blue-green to magenta. Small BIST ratified retinal ganglion cells oppose input from the S cones to input from the L and M cones. This is often thought to correspond to blue-yellow opponency, but actually runs along a color axis from yellow-green to violet. Visual information is then sent to the brain from retinal ganglion cells via the optic nerve to the optic chiasma, a point where the two optic nerves meet, and information from the temporal, contralateral, visual field crosses to the other side of the brain. After the optic chiasma, the visual tracts are referred to as the optic tracts, which enter the thalamus to synapse at the lateral geniculate nucleus, LGN. The lateral geniculate nucleus is divided into laminae, zones, of which there are three types: the M laminae, consisting primarily of M cells, the P laminae, consisting primarily of P cells, and the coniocellular laminae. M and P cells receive relatively balanced input from both L and M cones throughout most of the retina, although this seems to not be the case at the fovea, with midget cells synapsing in the P laminae. The coniocellular laminae receive axons from the small BIST ratified ganglion cells. After synapsing at the LGN, the visual tract continues on back to the primary visual cortex, V1, located at the back of the brain within the occipital lobe. Within V1 there is a distinct band, striation. This is also referred to as striate cortex, with other cortical visual regions referred to collectively as extrastriate cortex. It is at this stage that color processing becomes much more complicated. In V1 the simple three-color segregation begins to break down. Many cells in V1 respond to some parts of the spectrum better than others, but this color tuning is often different depending on the adaptation state of the visual system. A given cell that might respond best to long wavelength light if the light is relatively bright might then become responsive to all wavelengths if the stimulus is relatively dim. Because the color tuning of these cells is not stable, some believe that a different, relatively small, population of neurons in V1 is responsible for color vision. These specialized color cells often have receptive fields that can compute local cone ratios. Such double opponent cells were initially described in the goldfish retina by Nigel Daw. Their existence in primates was suggested by David H. Hubel and Torsten Wiesel and subsequently proven by Bevel Conway. As Margaret Livingstone and David Hubel showed, double opponent cells are clustered within localized regions of V1 called blobs, and are thought to come in two flavors, red-green and blue-yellow. Red-green cells compare the relative amounts of red-green in one part of a scene with the amount of red-green in an adjacent part of the scene, responding best to local color contrast, red next to green. Modeling studies have shown that double opponent cells are ideal candidates for the neural machinery of color constancy explained by Edwin H. Land in his Retinex theory. From the V1 blobs, color information is sent to cells in the second visual area, V2. The cells in V2 that are most strongly color-tuned are clustered in the thin stripes that, like the blobs in V1, stain for the enzyme cytochrome oxidase. Separating the thin stripes are interstripes and thick stripes, which seem to be concerned with other visual information like motion in high-resolution form. Neurons in V2 then synapse onto cells in the extended V4. This area includes not only V4, but two other areas in the posterior inferior temporal cortex, anterior to area V3, the dorsal posterior inferior temporal cortex, and posterior teo. Area V4 was initially suggested by Semir Zeki to be exclusively dedicated to color, but this is now thought to be incorrect. In particular, the presence in V4 of orientation-selective cells led to the view that V4 is involved in processing both color and form associated with color. Color processing in the extended V4 occurs in millimeter-sized color modules called globs. This is the first part of the brain in which color is processed in terms of the full range of hues found in color space. Anatomical studies have shown that neurons in extended V4 provide input to the inferior temporal lobe. IT cortex is thought to integrate color information with shape and form, although it has been difficult to define the appropriate criteria for this claim. Despite this murkiness, it has been useful to characterize this pathway, V1 V2 V4 IT, as the ventral stream or the what pathway, distinguished from the dorsal stream, where pathway, that is thought to analyze motion, among many other features. Subjectivity of color perception. Nothing categorically distinguishes the visible spectrum of electromagnetic radiation from invisible portions of the broader spectrum. In this sense, color is not a property of electromagnetic radiation, but a feature of visual perception by an observer. Furthermore, there is an arbitrary mapping between wavelengths of light in the visual spectrum and human experiences of color. Although most people are assumed to have the same mapping, the philosopher John Locke recognized that alternatives are possible, and described one such hypothetical case with the inverted spectrum thought experiment. For example, someone with an inverted spectrum might experience green while seeing red, 700 nm, light, and experience red while seeing green, 530 nm, light. Synesthesia, or idesthesia, provides some atypical but illuminating examples of subjective color experience triggered by input that is not even light, such as sounds or shapes. The possibility of a clean dissociation between color experience from properties of the world reveals that color is a subjective psychological phenomenon. The Himba people have been found to categorize colors differently from most Euro-Americans and are able to easily distinguish close shades of green, barely discernible for most people. The Himba have created a very different color scheme which divides the spectrum to dark shades, Zuzu in Himba, very light, Vapa, vivid blue and green, Buru, and dry colors as an adaptation to their specific way of life. Perception of color depends heavily on the context in which the perceived object is presented. For example, a white page under blue, pink, or purple light will reflect mostly blue, pink, or purple light to the eye, respectively. The brain, however, compensates for the effect of lighting, based on the color shift of surrounding objects, and is more likely to interpret the page as white under all three conditions, a phenomenon known as color constancy. In other animal species, many species can see light with frequencies outside the human visible spectrum. Bees and many other insects can detect ultraviolet light, which helps them to find nectar in flowers. Plant species that depend on insect pollination may owe reproductive success to ultraviolet colors and patterns rather than how colorful they appear to humans. Birds, too, can see into the ultraviolet, 300–400 nm, and some have sex-dependent markings on their plumage that are visible only in the ultraviolet range. Many animals that can see into the ultraviolet range, however, cannot see red light or any other reddish wavelengths. For example, bees' visible spectrum ends at about 590 nm, just before the orange wavelengths start. Birds, however, can see some red wavelengths, although not as far into the light spectrum as humans. It is an incorrect popular belief that the common goldfish is the only animal that can see both infrared and ultraviolet light. Their color vision extends into the ultraviolet but not the infrared. The basis for this variation is the number of cone types that differ between species. Mammals in general have color vision of a limited type, and usually have red-green color blindness, with only 2 types of cones. Humans, some primates, and some marsupials see an extended range of colors, but only by comparison with other mammals. Most non-mammalian vertebrate species distinguish different colors at least as well as humans, and many species of birds, fish, reptiles, and amphibians, and some invertebrates, have more than 3 cone types and probably superior color vision to humans. In most Catarrhini, Old World monkeys and apes primates closely related to humans, there are 3 types of color receptors, known as cone cells, resulting in trichromatic color vision. These primates, like humans, are known as trichromat. Many other primates, including New World monkeys, and other mammals are dichromat, which is the general color vision state for mammals that are active during the day, i.e., felines, canines, ungulates. Nocturnal mammals may have little or no color vision. Trichromat non-primate mammals are rare. Many invertebrates have color vision. Honeybees and bumblebees have trichromatic color vision which is insensitive to red but sensitive to ultraviolet. Osmia rufa, for example, possess a trichromatic color system, which they use in foraging for pollen from flowers. In view of the importance of color vision to bees, one might expect these receptor sensitivities to reflect their specific visual ecology, for example the types of flowers that they visit. However, the main groups of hymenopteran insects excluding ants, i.e., bees, wasps, and sawflies, mostly have 3 types of photoreceptor, with spectral sensitivities similar to the honeybees. Papilio butterflies possess 6 types of photoreceptors and may have pentachromatic vision. The most complex color vision system in the animal kingdom has been found in stomatopods, such as the mantis shrimp, with up to 12 spectral receptor types thought to work as multiple dichromatic units. Vertebrate animals such as tropical fish and birds sometimes have more complex color vision systems than humans, thus the many subtle colors they exhibit generally serve as direct signals for other fish or birds, and not to signal mammals. In bird vision, tetrachromacy is achieved through up to 4 cone types, depending on species. Each single cone contains one of the 4 main types of vertebrate cone photopigment, LWS/MWS, RH2, SWS2 and SWS1, and has a colored oil droplet in its inner segment. Brightly colored oil droplets inside the cones shift or narrow the spectral sensitivity of the cell. It has been suggested that it is likely that pigeons are pentachromats. Reptiles and amphibians also have 4 cone types, occasionally 5, and probably see at least the same number of colors that humans do, or perhaps more. In addition, some nocturnal geckos have the capability of seeing color in dim light. In the evolution of mammals, segments of color vision were lost, then for a few species of primates, regained by gene duplication. Eutherian mammals other than primates, for example, dogs, mammalian farm animals, generally have less effective 2-receptor, dichromatic, color perception systems, which distinguish blue, green, and yellow but cannot distinguish oranges and reds. There is some evidence that a few mammals, such as cats, have redeveloped the ability to distinguish longer wavelength colors, in at least a limited way, via 1 amino acid mutations in opsin genes. The adaptation to see reds is particularly important for primate mammals, since it leads to identification of fruits, and also newly sprouting reddish leaves, which are particularly nutritious. However, even among primates, full color vision differs between New World and Old World monkeys. Old World primates, including monkeys and all apes, have vision similar to humans. New World monkeys may or may not have color sensitivity at this level. In most species, males are dichromat, and about 60% of females are trichromat, but the owl monkeys are cone monochromat, and both sexes of howler monkeys are trichromat. Visual sensitivity differences between males and females in a single species is due to the gene for yellow-green sensitive opsin protein, which confers ability to differentiate red from green, residing on the X-sex chromosome. Several marsupials such as the fat-tailed dunnart, Sminthypsis crassicaudata, have been shown to have trichromatic color vision. Marine mammals, adapted for low-light vision, have only a single cone type and are thus monochromat. Evolution. Color perception mechanisms are highly dependent on evolutionary factors, of which the most prominent is thought to be satisfactory recognition of food sources. In herbivorous primates, color perception is essential for finding proper, immature, leaves. In hummingbirds, particular flower types are often recognized by color as well. On the other hand, nocturnal mammals have less developed color vision, since adequate light is needed for cones to function properly. There is evidence that ultraviolet light plays a part in color perception in many branches of the animal kingdom, especially insects. In general, the optical spectrum encompasses the most common electronic transitions in matter and is therefore the most useful for collecting information about the environment. The evolution of trichromatic color vision in primates occurred as the ancestors of modern monkeys, apes, and humans switched to diurnal, daytime, activity and began consuming fruits and leaves from flowering plants. Color vision, with UV discrimination, is also present in a number of arthropods, the only terrestrial animals besides the vertebrates to possess this trait. Some animals can distinguish colors in the ultraviolet spectrum. The UV spectrum falls outside the human visible range, except for some cataract surgery patients. Birds, turtles, lizards, many fish, and some rodents have UV receptors in their retinas. These animals can see the UV patterns found on flowers and other wildlife that are otherwise invisible to the human eye. Ultraviolet vision is an especially important adaptation in birds. It allows birds to spot small prey from a distance, navigate, avoid predators, and forage while flying at high speeds. Birds also utilize their broad-spectrum vision to recognize other birds, and in sexual selection. Mathematics of color perception. A physical color is a combination of pure spectral colors, in the visible range. Since there are, in principle, infinitely many distinct spectral colors, the set of all physical colors may be thought of as an infinite-dimensional vector space, in fact a Hilbert space. We call this space color. More technically, the space of physical colors may be considered to be the mathematical cone over the simplex whose vertices are the spectral colors, with white at the centroid of the simplex, black at the apex of the cone, and the monochromatic color associated with any given vertex somewhere along the line from that vertex to the apex depending on its brightness. An element C of color is a function from the range of visible wavelengths considered as an interval of real numbers to the real numbers, assigning to each wavelength W in its intensity C, W. A humanly perceived color may be modeled as three numbers, the extents to which each of the three types of cones is stimulated. Thus a humanly perceived color may be thought of as a point in three-dimensional Euclidean space. We call this space R3-COLOR. Since each wavelength W stimulates each of the three types of cone cells to a known extent, these extents may be represented by three functions S, W, M, W, L, W, corresponding to the response of the S, M, and L cone cells, respectively. Finally, since a beam of light can be composed of many different wavelengths, To determine the extent to which a physical color C in color stimulates each cone cell, we must calculate the integral, with respect to W, over the interval, of C, W, S, W, of C, W, M, W, and of C, W, L, W. The triple of resulting numbers associates with each physical color C, which is an element in color, a particular perceived color, which is a single point in R3COLOR. This association is easily seen to be linear. It may also easily be seen that many different elements in the physical space color can all result in the same single perceived color in R3COLOR, so a perceived color is not unique to one physical color. Thus human color perception is determined by a specific, non-unique linear mapping from the infinite-dimensional Hilbert space color to the three-dimensional Euclidean space R3COLOR. Technically, the image of the mathematical cone over the simplex whose vertices are the spectral colors, by this linear mapping, is also a mathematical cone in R3COLOR. Moving directly away from the vertex of this cone represents maintaining the same chromaticity while increasing its intensity. Taking a cross-section of this cone yields a 2D chromaticity space. Both the 3D cone and its projection or cross-section are convex sets, that is, any mixture of spectral colors is also a color. In practice, it would be quite difficult to physiologically measure an individual's 3-cone responses to various physical color stimuli. Instead, a psychophysical approach is taken. 3 specific benchmark test lights are typically used, let us call them S, M, and L. To calibrate human perceptual space, scientists allowed human subjects to try to match any physical color by turning dials to create specific combinations of intensities, is, IM, IL, for the S, M, and L lights, RESP, until a match was found. This needed only to be done for physical colors that are spectral, since a linear combination of spectral colors will be matched by the same linear combination of their, is, IM, IL, matches. Note that in practice, often at least one of S, M, L would have to be added with some intensity to the physical test color, and that combination matched by a linear combination of the remaining 2 lights. Across different individuals, without color blindness, the matchings turned out to be nearly identical. By considering all the resulting combinations of intensities, is, IM, IL, as a subset of 3 space, a model for human perceptual color space is formed. Note that when one of S, M, L had to be added to the test color, its intensity was counted as negative. Again, this turns out to be a mathematical cone, not a quadric, but rather all rays through the origin in 3 space passing through a certain convex set. Again, this cone has the property that moving directly away from the origin corresponds to increasing the intensity of the S, M, L lights proportionately. Again, a cross-section of this cone is a planar shape that is, by definition, the space of chromaticities, informally, distinct colors. One particular such cross-section, corresponding to constant x and y and z of the CIE 1931 color space, gives the CIE chromaticity diagram. This system implies that for any hue or non-spectral color not on the boundary of the chromaticity diagram, there are infinitely many distinct physical spectra that are all perceived as that hue or color. So, in general there is no such thing as the combination of spectral colors that we perceive as, say, a specific version of tan. Instead there are infinitely many possibilities that produce that exact color. The boundary colors that are pure spectral colors can be perceived only in response to light that is purely at the associated wavelength, while the boundary colors on the line of purples can each only be generated by a specific ratio of the pure violet and the pure red at the ends of the visible spectral colors. The CIE chromaticity diagram is horseshoe-shaped, with its curved edge corresponding to all spectral colors, the spectral locus, and the remaining straight edge corresponding to the most saturated purples, mixtures of red and violet. Chromatic adaptation. In color science, chromatic adaptation is the estimation of the representation of an object under a different light source from the one in which it was recorded. A common application is to find a chromatic adaptation transform, CAT, that will make the recording of a neutral object appear neutral, color balance, while keeping other colors also looking realistic. For example, chromatic adaptation transforms are used when converting images between ICC profiles with different white points. Adobe Photoshop, for example, uses the Bradford CAT. In color vision, chromatic adaptation refers to color constancy, the ability of the visual system to preserve the appearance of an object under a wide range of light sources.
Please subscribe and thanks for watching.